MS Ramaiah University of Applied Sciences.
Hi students, today we are going to learn about section cutting techniques. Aim of the experiment is to demonstrate section cutting and thereby enabling to prepare the slides for focusing and to study the microscopical characters. Apparatus and reagents required: watch glass, test tube holder, test tube stand, a sharp razor blade, camel hairbrush, glass slides, cover slips, a beaker with water, a dropper, filter paper or blotting paper, drug sample, chloral hydrate solution, Bunsen burner. Procedure. The first step in section cutting is selection of drug sample for section cutting. Selection of appropriate size, shape and part of the crude drug sample is very important in obtaining good sections. In case of stem or root, select a portion having a diameter of 3 to 5 mm and length of 25 cm. A sample shorter in length will be difficult to hold, and a sample thicker in diameter may give rise to thick sections. In case of leaf, young leaf is used because it has thin midrib and lamina, hence it is more convenient to obtain thin sections. Step 2. Preparation of sample for sectioning. Take the sample in a test tube and add sufficient chloral hydrate solution or water so that sample remains submerged. Light the burner with matchbox or lighter. Boil the sample over Bunsen flame for few minutes. This will soften the hard drug and will help in obtaining fine sections. In case of leaf, boiling is not required since fresh leaves are used. Step 3. Sectioning. Hold the sample vertically between the first and thumb finger and move the blade back and forth from one end to the other end, obtaining thin slices. Take sufficient number of sections as all the section will not be very fine and uniform. Transfer the section to a watch glass with the help of a brush. Reject thick and oblique sections. Precaution. Before taking section, ensure that the blade is having enough amount of water on its edge. If a dry blade is used, it shall entrap air bubbles in the section which are difficult to remove. Report. Section cutting of the given sample was performed.
Hi students, title of the experiment is section cutting of plant material. Aim of the experiment is to learn about section cutting and to prepare thin sections of the selected plant material. Chemicals required for this experiment are chloral hydrate. Chloral hydrate is a clearing solution used to clear the sections from pigments and fatty material. Fluoroglucinol, concentrated hydrochloric acid. Fluoroglucinol and concentrated hydrochloric acid are used as a staining reagent to stain the lignin present in the section. Glycerin. Glycerin is used as a mount to mount the section onto a glass slide, and glycerin prevents the drying of the section for 1 or 2 days. The components required for this experiment are test tube stand, test tubes, matchbox, test tube holder, razor, glass slide, cover slip, watch glass, compound microscope, filter paper, and the selected plant material for preparing sections, distilled water and Bunsen burner. Now let us start the experiment. The first step in this experiment is preparation of sample for sectioning. If the selected plant material is hard and dried, like roots, seed, stem and fruits We have to boil the material in sufficient quantity of water so that it becomes softened and it is ready for sectioning. Suppose if the plant material is a leaf crude rug and if it is a dried leaf, then it should be boiled in water to make it soften. If the material is a fresh leaf, then boiling is not required. Sections can be prepared from the midrib region of the leaf. And preferably, young leaves are selected for section so that we will get thin sections of the leaf. The next step in the preparation of sample is boiling the selected plant material. So today I have selected the stem of a young herb. We will light the Bunsen burner, switch on the Bunsen burner, light the matchbox. Now the burner is on. Take a clean test tube. Add sufficient quantity of water, preferably distilled water, and add 2 to 3 pieces of the selected plant material. With the help of a test tube holder, clamp the test tube and we are going to boil the plant material. The plant material should be boiled until it becomes softened. So it will take nearly 30 to 40 minutes. If it is a harder part of the plant like hard root, it will take more than 1 hour for softening. Now almost 1 hour is over. Now we can put off the Bunsen burner, remove the plant material, place it on a test tube stand, remove the clamp and allow the material to cool and switch off the burner. Turn the knob off. Now after boiling, the material is transferred to a watch glass. And to another watch glass, add sufficient distilled water. Now open the new razor for sectioning. A considerable size of a considerable piece of the material is chosen and both the upper and lower surface of the material should be leveled so that uniform sections are obtained. Now I am leveling upper surface and the lower surface. Now both the surfaces are leveled. Now for sectioning, the material should be held between the thumb and index finger. So hold the material between the thumb and the index finger firmly and gently. We should not press the material. Now each time when we take section, both the material and the razor should be dipped in water. The reason is, if you take section using a dry razor, there will be a chance of entrapment of air bubble into the section, which is difficult to remove later. That's why each time you take section, both the material and the blade should be dipped in water. So hold the material gently. Now move the razor front and back. Now we have taken 3 to 4 sections. Now, with the help of camel hairbrush, preferably 0 number, dip the brush in water and transfer these sections into another watch glass containing water. Similarly, sufficient number of sections should be taken, and from the sections taken, thin complete sections are selected for staining process. This completes the experiment, section cutting of plant material. Thank you.
At MSR UAS, we spark your imagination unlike any other.